Welcome to our tutorial to querying gene expression datasets in braintrawler. We begin by selecting the human or mouse brain to enter the application. The first thing you will see is braintrawler's Browse Database view, which gives you information on all available data. Clicking on a dataset reveals associated metadata on the right. Now, let's proceed to the Gene Expression Query page by clicking Explore Regions and Gene Expression Query. On this page, you will see a 3D and 2D representation of the brain. First, we will choose a dataset to query. Here, we go for the Allen Human Brain Atlas dataset. Next, we can decide where in the brain we want to search for high gene expression. We can either choose brain regions from the Allen Human Brain Reference Atlas, as we do now, or create a manual spatial selection by drawing in the 2D slice view. The brain region we selected is now highlighted in the renderer. Next, we can choose between searching for high or specific expression in this area. Let's look for highly expressed genes. This executes a query that will give us a list of all genes in the dataset, ranked by their expression in the query region. Each gene can be visualized by clicking the eye icon in the list. To get a better understanding of the expression levels of a gene, you can also investigate the expression profile in the Gene subtab. You can find additional information on the braintrawler help page. Thanks for watching.